Ms. Middleton, I'd like to turn now to the fractionation of human factor VIII by the polyelectrolyte method. I won't take you to your statement, but you say in it that shortly after, or relatively shortly after joining Spaywood, you were seconded to BPL and you subsequently spent some time in Paris as well at the CNTS lab. I think that was from April 1981. We heard in the presentation about how there was some optimism about the product in 1981, which gave way fairly quickly to a more pessimistic view of the prospects of the product. Can I ask, you have said in your statement that yields were variable and that problems soon became apparent in the product. Could you explain what those problems were and how quickly they became apparent to you and to others?
Well, as you probably know, blood clotting is quite a complex process. It's a cascade. That's to make the blood clot. There are also a lot of mechanisms in there to stop blood clotting when it's not supposed to. So it's quite a complex system to deal with. And if you are making factor VIII, purifying it, depending on the starting material, you have plasma, which is in various... Once you collect plasma or blood from the body, it's immediately not as stable as it was when it was in the body, because you've exposed it to surfaces, and you've generally exposed it to the air, and you've generally changed the profile of it, and the inhibitors to blood clotting and the procoagulant. So particularly with the polyelectrolyte, the factor VIII, it proved to be very difficult to control the clotting, if you like, because the factor VIII was probably becoming activated during the process. And how activated it became depended on the quality of the starting material quite often. But it did result with huge variability trying to control the process. And it proved, in my hands anyway, very difficult to control it with any of the sort of different parameters that I tried. So you absorb onto the polyelectrolyte and then you wash off. That's essentially what you have to do. By controlling the way you absorb it, or the things you add to it to keep it stable, the way you elute it off, can all affect what happens in terms of the stability of the final product. And what it proved to be relatively easy, you could elute things off, and you could measure a factor VIII level that seemingly was high, remembering that the assays themselves could sometimes not discriminate as to whether you'd got an activated system or not. And then as you stored it and let it stand, the activity dropped off because the factor VIII was becoming activated and lost. So it proved very difficult to control. And that, in essence, was confirmed because when we made factor VIII for sequencing, that was Professor Tuddenham's project, he had to use a very serious antiproteolytic agent, it was actually nerve gas, to stop the clotting factors, prevent activation of the clotting cascade in order to get pure factor VIII at the end of it.
If I can just interject there, a couple of things arise from that. The variability of the product, does that make it hard to translate from a laboratory-based product on which you can do your experiments and test for potential into a mass-produced product which can actually be marketed?
The main thing that you have to remember when you're producing any product is quality, safety, and efficacy. If you're getting variability yields and you don't know why, then you haven't got quality, and not the safety either.
The process that you were talking about with Dr. Tuddenham there, I'm going to come on to recombinant shortly. But there is an article that he has written that I know that you have seen. I'm not going to bring it up, but the reference is HSOC 0022968. And he is describing how he worked with you and with others in order to try to purify the factor VIII that he could then go on to use in gene sequencing, which would then give rise to recombinant products. And he said that by 1982, he had devised a multi-step procedure which led to that, in cooperation with others, I should emphasize. And he said the bulk processing capacity of polyelectrolyte, combined with the exquisite specificity of monoclonal antibodies, overcame all remaining problems, although Francis Rotblat and Don O'Brien in my laboratory found that they needed to use large amounts of highly poisonous nerve gas type enzyme inhibitors to keep the factor VIII stable. So that is what you were referring to a moment ago.
Yes.
I don't think it needs to be stated why using a nerve agent as part of a product.
Not suitable for a therapeutic product. No.
Exactly. Thinking back now, are you able to put a timescale on when you became aware that these problems were going to be insurmountable in terms of bringing a product to market?
Well, I think I became aware of it when I was actually at Elstree.
This is 1980 to 1981.
Yes, if that's when it was, yes. And I spent a couple of months after that in Orsay in Paris, and we had the same problems. We couldn't get the thing to be stable. You know, one day you got something what you thought was 75% yield, and then if you left it on the bench, it had gone down to 25%. So it was really not looking very viable from the stage at Elstree.
You were in Paris from April 1981, according to paragraph 17 of your statement, for a few months.
Yeah, for a few months, yes.
But by that stage, do you think that you were aware that this wasn't really going to be viable?
Well, it just never seemed to, no. I thought.
I'm just going to ask for a document to be brought up on screen. It's BPLL 00016007_018. Please, Sumik. Sorry, BPLL 00160007_018. This is a letter which is written to Mr. Heath from Dr. Elaine in Paris. I know that you have seen this in the files we've provided you with. He says, Dear David, I enclose a summary of the results of PE5 phase 1, which has worked out very well. We are presently doing some additional experiments on filtration and freeze drying that looks very promising. The loss in yield seems quite reasonable. I've done all the background work for the next step, and the raw material is now being prepared. I'm confident that step 2 will be as successful as step 1. So that's Dr. Allaine's view in this letter on the 27th of May 1981.
Yes.
Now, I'm cautious about reading too much into one letter in a snapshot in time. But at around that time, had you already doubts developing in your mind?
I did. And obviously, Jean-Pierre Allain and I worked together on the preparation in stage, I think it was step 2 in Paris. And we had the same problems as I'd had at BPL. So I think his letter was optimistic.
His letter, of course, is referring to him at the end of step 1.
Yes.
You subsequently worked with step 2 and was disappointed.
And we never really went to step 3, I don't think.
We heard earlier, I believe you were in the room for it, about Dr. Tuddenham's lecture from some point in 1981 in which he expressed some optimism. There is also a paper, I won't bring it up, but BPLL 00160007_026. The authors are Tuddenham, Lane, Rotsblatt, Johnson, Snape, you and Kernoff, response to infusions of polyelectrolyte fractionated humanate concentrate
Yes.
in human haemophilia A and
Yeah.
von Willebrand's disease.
Yes.
Both of those, both the lecture and the paper, express some hope
Yeah.
for the method.
Yes. We made a batch at BPL with carefully collected, in fact, I think it was almost fresh cryoprecipitate from very good quality specifically for those clinical trials, because it was done in 4 patients, I think, 3 haemophiliacs and 1 von Willebrand.
Yes.
Yes. And specifically for that, it was made in the clean room at BPL and specifically for that experiment. And that showed that the factor 8 that we got was good recovery, which suggests it was quite good factor 8, and that it didn't work in von Willebrand's disease, which is what we might have expected because of the nature of the protein at that point.
So an example of how, when it is done in very closely controlled circumstances, it could work, but that doesn't
Yes,
answer
and that was probably due to the fact that the cryoprecipitate was very good quality. The product I was working with at BPL was mostly freeze-dried product that had failed quality control for some reason, mostly due to pyrogen or bacteria, well, pyrogen really, or something of that order. So I worked on that, which had been freeze-dried and was not terribly good quality. The cryo that I worked with on that product was possibly fresh and probably never been even frozen. It was probably made straight from that. So if you had perfectly good quality cryo, then maybe it would work.
You also wrote a short paper on viral inactivation, which is dated October 1983.
Yeah.
I won't bring it up. It's ISPN 40409_009.
Virus separation.
But sorry, viral separation.
Yes.
My error. If I may summarise the paper, it's that the in vitro experiment that you did showed viral separation.
Yeah.
But as per Dr. Tuddenham's lecture, there was a need to look at that in clinical trials if that were to be proved to be safe and efficacious.
Yes, we showed that in Professor Thomas's lab. We added hepatitis to the concentrate and then worked it over the polyelectrolyte, and that showed that there was no absorption of the virus to the ion exchange resin.
Meaning that the ultimate product would be free of hepatitis B.
Would be separated from it, yes.
And was there an expectation that it would also clear NA and B hepatitis?
Possibly for the same reason.
You say possibly, that of course couldn't be tested for.
No.
Hence the need for clinical trials. We'll come back to the clinical trial in a second. Before I do, we know that you developed your concerns about how viable this was going to be as a product. Did you share those concerns with Mr. Heath?
Yes.
We've seen some of the things that he was saying in 1981. To give you just one example from July 1981, the Human Protein Plan. He said, quotes, recent production yields, clinical trials and hepatitis tests have adequately demonstrated that the major protein, factor 8C, can be produced economically, is very efficacious, and is most probably free of hepatitis infectivity. That is from July 1981. In your view, was that a claim that was justified?
I think it was optimistic at that point.
This was at a point in time when Mr. Heath and Spayward were seeking a £4 to £5 million investment, half of which was coming from public funds. In that context, Do you think that that claim was one that could reasonably have been made?
With hindsight, was perhaps a bit economical with the truth.
Were you involved in any way in the phrasing?
No, no.
If you had been involved, what would you have said to Mr. Heath about it at the time?
Well, I would have said, I would have suggested that he moderated his language.
Sorry.
Well, I think I should say something about David, because he was very entrepreneurial, and he was entrepreneurial from very early. He took on a porcine factor 8. Why on earth would he do that? He was a pharmacist. It was a very strange thing to do, but he saw the potential in it. He took on the polyelectrolyte fractionation to improve that porcine factor 8, which again was very entrepreneurial. And obviously the recombinant factor 8 came from him, and he worked on it. But the trouble was that he was not very good at man management, and people let him down in some of the things they did. And that was a shame. And he also ended up, he could very easily, because he was the way he was, put people's backs up. And that was certainly the case with the bioproducts laboratory, because of the different cultures. Bioproducts was there to make blood products and get as much as possible out for the population. David was constantly looking into the future and trying to raise money in those days was difficult, and therefore you tend to overpromise, or he was overpromising. The porcine factor 8 was working very well, mainly because I think a lot of the clotting that happened and the problems with clotting happened before the plasma ever got from the abattoir to what we were working with, and we were left, and the yields were probably pretty dreadful from porcine blood. But it was easier in that sense because we had, but with factor 8, you were so totally dependent on fresh frozen, good quality human plasma, human blood, that you had to be so careful about the yield. So he was optimistic because of the porcine was going well. He maybe thought that the human should be going well because the porcine was. And that was probably his reasons for doing it.
But by July 1981, he was aware, was he, of your reservations?
He knew, but he probably thought I was just being, slightly negative on the subject. Quite likely.
You were the chief scientist of Spayward, and he was a pharmacist by trade, as I understand it.
Yes, he was.
The clinical trial of Mono C, the factor 8 product, were you involved in putting the application for the clinical trial together?
Well, I must have been, but I don't remember doing it. But we must have done it.
We know that it went in at some point in 1982, and we also know that progress on the product had slowed by that stage. What was the purpose of having a clinical trial in those circumstances?
I think it was basically to check, to see whether it was safe and whether the concept of factor 8C, which is what it was, the clotting activity, as opposed to the complex, which circulates in plasma with the von Willebrand factor sort of attached to it, or that was what was thought, whether it would survive like that in the circulation.
Was there any feasible prospect, as of 1982, of bringing this to a product that could be placed on the market?
At the time of the clinical trial?
Yes.
Maybe in some, I don't know how to answer that question.
Let me...
As best you can.
As best I can. Because the question is, why did you do a clinical trial?
That's no smoke and mirrors here. That's the next
That's the question.
If you're in the circumstances where it is unlikely that this will ever be made into a
Well, it was made at the Bioproducts Laboratory, that product. So they must have thought something positive about it at that stage, in spite of the difficulties that we had with it. So maybe they thought that they should check that it would work, because it was a different entity. So maybe that, things might work out. Maybe I was being too negative about it. Maybe it was possible to make it work. But the other question that needed to be answered is, would it actually work in the circulation? Never mind whether because if you can get the production process to work, which I couldn't, but other people might have got it to work, would factor 8C on its own last in the circulation? And yes, it did.
Do I take it from your answer that you were trying now to help us by looking back and thinking what the thinking might have been, but you're not able to say what you yourself thought at the time?
Yes.
I should say, out of fairness, that we looked at the 1983 to 1985 business plan,
Yeah.
and as we discussed during the presentation, the door was not being closed absolutely on factor 8 through polyelectrolyte fractionation, although there were concerns about how feasible it was going to be. We know that the initial application was turned down, and I referred to a meeting that you attended on the 2nd of June 1983. And we could have that on screen, please, Shomik. It's IPSN.
That wasn't the same product.
If we bring it up on screen, perhaps you can assist us with it. IPSN 40165_009, please.
Yeah.
So this is a meeting on the 2nd of June,
Yeah.
1983. The subject, clinical trial certificate for mono 8C,
Yeah.
and then source of cryoprecipitate.
Which was this alpha cryoprecipitate, which was being bought in 100 kilo lots, in large lots, as a frozen cryoprecipitate to try to make factor 8C out of. It wasn't that material that went into the patients. The product that went into patients came from Elstree.
Yes, I think we're at slightly cross
Right, okay.
So the paper that I referred you to earlier, that you co-wrote with Dr. Lane, etc.,
Yes.
and Dr. Tuddenham, that was a product that was made in Elstree,
yes.
and that was used, as you said, on 3 patients with
And
haemophilia
one
A
von
and 1 von Willebrand's patient.
Yeah.
This is different. This is the application for the clinical trial, which was made at some point in 1982.
Yeah.
That was what I was discussing with you a moment ago.
Yes.
Why it was that there was an attempt to have a clinical trial at that stage.
From this, yes.
Does that alter your assessment? Any of your answers now that we're clearer about what we're talking about?
I don't see why it should alter your answers, but just that we now know you're talking about, we're quite clear which trial you were now talking about.
Yes. I recall this, and as you know, in the plans, there was the idea of building a facility to make human factor 8 from this frozen cryoprecipitate. I wasn't certain about it at all, but I'm not sure why we applied for a clinical trial certificate using this material.
The idea was that the material would come, the cryoprecipitate would come in from alpha, then you would fractionate it.
It was a frozen paste. that you resuspended. I did do some work with it. It was a frozen paste that was resuspended and then put over the polyelectrolyte.
I'd like to just look at what was said at this meeting. There was you and Anne Walton from Speywood. The note is by, the initials are EAW. Is that Anne Walton?
Yes.
Who was Anne Walton?
She was, she did some marketing, but she also focused in on regulatory affairs.
Was she a scientist?
Yes.
The participants from the DHSS are Dr. Fowler and Dr. Purvis.
Yeah.
And if we could just look at the note, and I'll read it through to you. Dr. Fowler was of the opinion that, despite the controversy surrounding US imports as a result of AIDS, our application will not be judged prejudicially by the CSM if we pursue it with alpha cryo cited as source material. There have been suggestions in certain quarters about the banning of importation of all US blood products, but the impracticality of this is recognised by those who are well informed in this area, and a ban does not, therefore, seem likely. An application based on alpha cryo would, or should, be judged solely on its scientific merit. The possibility of leaving an application open-ended with respect to source material was discussed and dismissed as unacceptable. We were advised that if a change in source material is desired, the application might proceed more easily if the licensing of the import of the cryo were included in the CTC application. The responsibility for the quality of the raw material would then be entirely Speywood's, and in addition, the cryo would then be licensed for importation only for the purpose covered by the CTC. If we go over to the next page, the possible courses of action are discussed. I won't go through that, but it says in conclusion, it appears that the use of US cryo will not prejudice our case with the licensing authorities, and therefore our choice of source material can be based on commercial and scientific grounds. A couple of points about that. Paragraph 1, and the discussion of the potential ban on US blood products and the recognition that that was impractical. Are you able to help us with who was saying that it would be impractical to ban US blood products?
No.
Would you or Anne Walton have been in a position to make that observation, or do you think it's more likely to have come from the DHSS officials?
I think it's more likely to have come from the DHSS officials.
The references to the source material choice being based on commercial and scientific grounds, as opposed to what other grounds?
As opposed, presumably, to citing the source of the raw material. So, in other words, it's coming from the US. I don't know what else.
Are you able to assist us any further now about the contents of that meeting and what was discussed at it? Can you remember it at all?
I can't remember it. I can't remember it very well at all, no.
Do you know why it was that you were asked to go from Speywood?
Well, Anne would have set it up, and I would have gone along because of being, whenever that meeting was, still being chief scientist of Speywood, with the most experience for the human factor VIII.
Can you help us with whether or not a further application was made for a clinical trial certificate on that product?
I don't think it was.
Do you know why it wasn't pursued?
No. Well, yes, I probably do, because I don't think there was anywhere to make it.
The facility that had been planned wasn't built?
No. And I think at one stage it might even have been thought you could share the facility with porcine, but that became a no-goer because of virus concerns, and it had to be separate.
That is something we saw in the business plan about the
Yeah.
DHSS no longer allowing a
Yeah.
multi-purpose site. Looking back on human factor VIII and polyelectrolyte fractionation, was this always a project that was ultimately bound to fail, given the knowledge and equipment and availability of material in the early 1980s, or were there any missed opportunities to have developed it further?
Sorry, do you mean with respect to human factor VIII?
Yes.
I don't think it was going to fly. It worked very nicely for porcine factor VIII.
But not for human.
But not for human.
On recombinants, I'm going to take this very briefly, if I may. Your role was important but limited in helping to provide the first step of the purification process,
Yeah.
and then passing that on to Dr. Tuddenham and his team to work on thereafter.
Yes.
Mr. Heath's view that we heard earlier was that this was something of a tragic failure by the UK to capitalise upon the work that was done, because ultimately the work was taken forward by GeneTech, an American company, and subsequently by other American pharmaceutical companies as well.
Yeah.
Could I ask for your opinion on that view from Mr. Heath, and in particular upon the question of whether, if other steps had been taken, it would have led to a quicker development of recombinant products, or just a development by a British firm as opposed to an American firm?
I think what happened with the recombinant factor VIII was that, as I understood it, there was a certain amount of money brought forward by Prutec and BTG, and that was sort of that money was dispersed around various Oxford facilities to try to clone the gene, to sequence it first, purify it, etc. And none of that program really worked out. The only bit of that program that worked out was Ted Tuddenham's purification. The UK didn't really have the technology at the time. In a paper that Ted Tuddenham wrote, he said that they had, and I recall that they did, they interviewed Genentech, Genetics Institute, with another American company, and Celtech in this country, after Ted had presented his work on purification. And Celtech in this country was at the stage of making protein, I think it was rennin, in bacteria. Factor VIII was a big protein. Nobody knew what the structure was. It needed to be done in mammalian cells. David had actually written into the original agreement that if it was mammalian cells, then the UK manufacturing rights would stay here. So he was astute enough to realise that, and they realised that it was never going to be a bacteria. And the Oxford people who were working on the project were working in yeast, which is not terribly much better for a big protein like factor VIII. There was not much known about the structure either. And I do recall that after some had been purified, it was sent to ICRF, but Mike Waterfield, who was going to do some sequencing, couldn't get time on the sequencer. So when it went over to Genentech, they had a whole department with several people, sequencers dedicated to the project. It was just completely different. So going back to your original question, which I've probably not answered, I don't know, but it came down to the fact that I think the funding from Prutec and BTG was not used in a very constructive way, and that goes back to what I said originally about David is that he gave responsibility to people for doing things, and the person that stood out was Ted Tuddenham. The rest of them didn't really have the technology to do it. And Prutec and BTG got a bit upset about that, and that's when the trouble came, and David was removed and put to one side. He wasn't terribly popular with a lot of people because of his sort of rather, what should we say, his sort of, his energetic sort of, I can't think of the right word, but he was passionate about what he wanted, but that passion tended to make him promise too much and achieve and not achieve it. And I don't think that made him a very good source for somebody to invest in. I don't think they were very happy with it.
Thank you. Those are the questions that I have for you. So may I suggest a few minutes break while we allow the call participants? I don't anticipate there will be many questions from them. I received a great deal of assistance in advance, Ms. Middleton.
Well, we'll see, because the questioning may have prompted some questions. What we always do, I don't know if you've watched any of the inquiry at all before you came, but what we always do is we take a break when a witness has finished the evidence which the inquiry counsel has to ask, to allow the core participants, they come from all different perspectives, to ask questions through counsel of the witness. And that way we make sure that we try to collect as many different questions, answering different viewpoints as there may be. It does mean you've got to wait a little bit longer. I can't promise you how long, but we'll say 15 minutes and come back at 4:45, and we'll see then what questions there are, if there are any.
Okay.
So 4:45.
Right.